In this video, we're going to talk about fungi in the phylum Zygomycota, which are also referred to as the conjugated fungi. The majority of fungi in Zygomycota are saprobes, and remember that saprobes are the decomposers, which break down dead and decaying organic matter into its smaller components. Some Zygomycota fungi are parasites, and they specifically parasitize insects. The thallus of Zygomycota are aseptate, and they have haploid nuclei in the vegetative state. And we'll talk about this more when we talk about the reproductive strategy of Zygomycota. Zygomycota fungi do have a commercial role. They are used to make synthetic steroid hormones. Steroid hormones include the sex hormones, so things like progesterone, estrogen, and testosterone. And cortisol, the stress hormone, is also a type of steroid hormone. Zygomycota might also be detrimental. So one example is bread mold. So this is the mold that forms on old bread that makes it go bad, and that is caused by the Zygomycota fungi. Zygomycota fungi usually reproduce asexually, and unfortunately this picture here doesn't show kind of a really important part of asexual reproduction in Zygomycota, and that is the production of sporangiospores. And what a sporangiospore is the structure that actually kind of grows out of the mycelia, and it has spores in it. So this is the sporangiospore. And then these little dots inside are the spores, and those spores get released into the environment from the sporangiospore. And that fuzzy looking appearance that you see when you look at bread mold, you're actually seeing the sporangiospores kind of sticking up and releasing spores. When conditions are unfavorable, the Zygomycota can actually switch over to sexual reproduction, and sexual reproduction in Zygomycota is known as conjugation, and it follows the same basic concepts that we covered in the video on fungal reproduction, but there are some specific structures involved that happens in the Zygomycota that maybe don't happen with the other phyla of fungi. So we're going to talk about that here. So now let's take a look at sexual reproduction in Zygomycota. Zygomycota have heterothallic plasmagamy. If you recall, plasmagamy is the first step of sexual reproduction in fungi. And plasmagamy is where the cell membrane and cell wall of two different mating type cells fuse, but the nuclei within the cells do not fuse. And remember that heterothallic, hetero means different or other, and thallic refers to the thallus or the mycelium or the hyphae that make up the thallus. So what happens is that there has to be two different fungi present, each with their own thallus, and each one of them is going to contribute one of the mating type cells towards the sexual reproduction encounter. So I'm going to draw out what's happening kind of in this area of the diagram. So essentially we have two different fungi. So I'll draw a mushroom here with its hyphae making up the mycelium, the thallus underground. And then we have a separate fungus with its own thallus, mycelium, hyphae underground as well. And we'll just say that the red fungus is going to be making the positive mating type cell, and the blue one is going to be making the negative mating type cell. So these fungi give off pheromones, which are kind of these like sex hormones that are a way for them to communicate with each other. And so the hyphae actually start to grow towards each other. And these hyphae, remember that they are aseptate. Meaning that they are these kind of long cells with multiple nuclei in them. Kind of like what you see over here, where we have these long cells with these multiple nuclei or little dots inside of them. As this process is occurring, each of the fungal hyphae that are kind of moving towards each other, they start to develop a gametangium. So gametangium is singular, gametangia is plural. And you can see that on this picture here. These little kind of bumps that are forming on the hyphae that are starting to move towards each other. So we can also draw that on our picture here. So the red forms a gametangium and the blue forms a gametangium. When the gametangia fuse together, they form a zygosporangium. So essentially each gametangium, they fuse together, but then they break off from the hypha that they originally formed from. And you can see that on this picture here. Now notice that because these are aseptate hyphae, there are multiple haploid nuclei that are located in these hyphae. So when that zygosporangium breaks off, we end up with this structure that has multiple haploid nuclei in it, some from the red positive mating type fungus and some from the blue negative mating type fungus. The second stage of sexual reproduction in fungi is karyogamy. During karyogamy, the nuclei that are inside the zygosporangium are going to fuse. And so ultimately, you're going to end up with a zygote that has multiple diploid nuclei in it. And I'm drawing the diploid nuclei as purple as a way to show that the nuclei, the blue and the red nuclei that I drew, are fusing together. But remember that in a diploid nucleus, you have these paired chromosomes. So each one of these kind of purple dots is actually representing a set of red chromosomes paired up with a set of blue chromosomes that have just come together and they're now being encased in the same nuclear membrane. But the chromosomes aren't fused together, just the nuclear membrane has fused together. So now we have these paired diploid chromosomes inside the zygote. So remember that this is the zygosporangium. With the haploid nuclei in it. And then down here we have the zygote, which has formed after karyogamy. And we have multiple diploid nuclei present. And you can see the zygote here on the diagram. The zygote is then going to go through the next stage of sexual reproduction, which is going to be meiosis. And meiosis happens inside a structure known as a sporangium. And so that zygote inside the sporangium is going to go through meiosis to form haploid spores. And remember that during meiosis, the paired chromosomes in the diploid nucleus get separated from each other, and they kind of get dealt out into the daughter cells in unique combinations. And so this is how we end up with an increase in genetic diversity through sexual reproduction, because we took the genetics of two different organisms, put them together, rearranged them, and then dealt them out, almost like dealing out cards after they have been shuffled. So meiosis ends up producing these cells that have a kind of a unique combination of the different chromosomes that we started out with in the gametangia. So some of the cells might have kind of a different arrangement of red and blue chromosomes in them. I need to fix the wording here a little bit where it says karyogamy produces zygospores. In reality, karyogamy produces zygotes with several diploid nuclei. Those zygotes then go through meiosis to form the zygospores. And the zygospores have haploid nuclei. Now it's important to note that the zygospores are protected by these thick coats, and that allows them to stay dormant until conditions improve. Once environmental conditions become favorable, the spores can then start to germinate. And during germination, which is our fourth step of sexual reproduction, the cells are going to go through mitosis to start making more cells in order to develop into a new fungal organism.